And hello, microbiology friends. Hope this lecture finds you doing well. I'm doing pretty good myself. And we're thinking about our exam here. So, because I got an appointment at 11, and it's actually about 9:20 right now in the morning on Wednesday morning, we're not going to have our Zoom call today. We're going to do that Friday. And it'll be a chance to talk about what's going on, see if Zoom is a way that we want to try and do more discussions. I'd love to actually have more discussions with you guys. And start thinking about how we're going to put together the exam. So we're going to finish up this chapter 5, on nutrition and environmental conditions for bacterial growth, and see where we go from there. There'll be I'll put together an announcement about a little assignment for you that kind of helps to push up exams. It's going to have you look at some papers or find an article that is relevant to microbiology, and particularly microbiology specifics of what we're talking about. And that'll be a thing that you can do, and you'll write a little one-page summary of it. So I'll get that to you. Well, we've been talking about some of the growth conditions that bacteria engage in, and the idea of the bacteria numbers in a beginning of an inoculation or an infection in what's called the lag phase, and then it goes up in the exponential logarithmic phase, and then we reach a condition in which bacterial death equals bacterial replication, and that is the plateau phase, the stationary phase. After a while, conditions become less favorable, and we start to have more deaths compared to the number of bacteria still alive and reproducing, and that is the decline phase, and that's when things begin to go down. I'm just looking at my notes to make sure I'm using the terms that they use. The death or the decline phase. That's it. Anyway, we then went into nutrition options. And one of the things I'm not sure that you're totally down with, have you guys had metabolism, things like the Krebs cycle? Do you know how energy is extracted from food? I don't know. So I thought I'd just throw a little bit of that in there. When we think about growing organisms, you and I, bacteria, anything, we need to have a couple of specific things in order to carry out growth. What we have to think about is we're going to be creating new molecules and building new biopolymers, making new bonds. And there are a couple of things that we have to have. One, you have to have some source of energy that is going to be eventually incorporated into ATP. Remember, we talked about enzymes can change shape and snap by adding that phosphate. Remember, negative charge. So we need to have an energy source. Now, we get that energy source from consuming food, and we'll get into some more metabolism later in the semester. Plants get that energy source actually from harvesting the energy from sunlight. Bacteria can do pretty much a variety of things. They can get the energy from sunlight, they can get energy from organic materials, they can even get energy from inorganic materials, which is pretty weird. Now, the next thing you need is a carbon source. That is, you're going to be building new molecules. We're carbon-based molecules, all those things we talked about, fats, proteins, carbohydrates, nucleic acids, all carbon-based. So you have to have a carbon source for new carbon. Now again, we get that from the same place that we get our energy, and that is from consuming carbon materials. If you're not eating carbon, you really need to think about your diet. Like, don't eat glass, don't eat metal, don't eat dirt, no. So we eat carbon, we consume that. That is then incorporated into the carbon molecules in our body. Now, plants are getting their carbon primarily from carbon dioxide in the atmosphere. This is photosynthesis. Bacteria are going to get their carbon from carbon dioxide or from organic materials. And so they have a couple of different ways that they can get their carbon as well. So, you have pretty much in the carbon source, you have two different options. You can either get it from CO2 or you can get it from complex organic molecules. Now, the last thing that may not be quite as obvious that what you need for survival, what you need for nutrition, is a source of electrons. Because as we're baking these polymers, we're creating new covalent bonds, and these covalent bonds require electrons. These are all going to be reduction reactions, and hopefully you remember this from chemistry. Oxidation is a loss of electrons, reduction is a gain of electrons. And so in order to carry out the reduction of carbon molecules into large polymers, you have to have a source of electrons. So these are the three things we need in terms of nutrition, in terms of things you need to grow. You need energy, you need electrons, and you need carbon. Now, we conveniently get that from one source, our chow. You don't have to work that way. There are organisms on the planet that don't work that way. They get their energy from one source, from the light or from chemical bonds. They get their electrons from inorganic molecules or from organic molecules. And then they get their carbon, pretty much everybody's getting their carbon from just two sources, either CO2 or organic chemicals. But that's the differences here. So I just was thinking that, the electrons are kind of a weird thing. Like, you need electrons to eat? Yeah, well, you do. You need electrons being brought in order to actually carry out metabolism. Now, plants actually are splitting electrons from water. And so when you are watering your plant, you actually are giving them a source of electrons. And they split the water, and then they give out oxygen, and they take the electrons they harvest from water, and they put that into their metabolic system. Now, another part of this game is that if you're going to be carrying out these chemical reactions, these reduction reactions, whenever somebody gets reduced, somebody else has to get oxidized. And so another common thing that we need to think about is that at some point you need a place to dump electrons. As you're consuming nutrients, whatever it is, you're going to be liberating electrons. Now you might be able to use those and sort of incorporate them, but generally we don't have a closed system like that. You're going to have something that you're going to dump those electrons onto. We'll get to that in just a moment. So the next part of the discussion of growth habit, we have food. We also have environmental conditions that are critical to bacterial growth. We know there are a whole range of conditions that are conducive to bacterial growth, that are optimal, and then there are a whole bunch of conditions that are not optimal for bacterial growth. And so if your goal is to reduce bacterial load, you would generate conditions that are not very favorable to bacteria. It's one of the reasons why surgical suites might be very cold, air conditioned really hard, keep people from sweating, but it also is slowing down bacterial growth. So temperature is a real critical part of this, and there are bacteria that grow optimally at body temperature. They're kind of designed to operate at around 37 degrees. And then there are bacteria that actually can grow and are optimized to grow at very low temperatures. There are bacteria that grow in the Arctic and on little Arctic shrimps and things in really icy cold water. And they're designed to grow in those conditions. In fact, they wouldn't grow at room temperature at 37 degrees. Another reason to think about this is that by putting stuff in the freezer or even in the refrigerator, you're going to slow down bacterial growth. Freezing is going to pretty much block bacterial growth of most of the bacteria that you're going to come across in your foods. There are also some very strange bacteria that have only in the past 30 years or so been discovered that are able to grow in really high temperatures. These are very heat tolerant, and they actually require high temperatures greater than 40 degrees, which is, you'd be running a pretty serious fever. And they are able to grow in these more, hot box-like conditions. And there are even a few that we refer to as hyperthermophiles that grow at temperatures greater than 70 degrees centigrade, which I'll just tell you it's hotter than the hottest water in your house. It is not quite boiling, but it's not bad coffee temperature. And I ain't talking no iced coffee. I'm talking hot coffee. So these are particularly strange bacteria. They actually have some really interesting properties in that their proteins don't denature at these high temperatures. All right, so just because I got them standing in front of me, I'm going to throw up some PowerPoints. I'm going to throw up See, if you're listening to the lectures, you might hear all kinds of amusing points. Maybe I'll start putting in little Easter egg things of, where's Dr. Galuski from? And you'll put that as an extra credit question. Where is he from? I'm from Beaumont, Texas. Not that anybody cares. But we got slammed by hurricanes routinely. I'm waiting for the projector to come up. Can you see it? Here it is. Let me kill the lights here just to get a little bit more contrast. So, these are some of the terms we use. This is the complicated slide that we finished up on, the nutrition options, where we got the idea of the source of energy, source of electrons, source of carbon. Break it down like that, set them up so you know the terms: phototrophs, chemotrophs, lithotrophs, organotrophs, autotrophs, heterotrophs. One of the things I think it's convenient is to think about what we are versus what a plant is, then some of the weird bacteria. So, environmental conditions. We've got things like sea ice, and sea ice is growing at a very cold conditions, and there can be bacteria actually in the ice that are thriving in these cold requiring, or these cold temperatures. So a couple of the vocabulary words. We've got psychrophiles. These are cold requiring. Cold tolerant are referred to as psychotrophs. You have troph being growth, phile meaning loves. And so that's always a way to think about this, is that a thermophile loves heat. A mesophile is growing in a moderate range, and they usually don't grow, in fact, they probably will die outside of this moderate range. 10 to 45 degrees, I gotta tell you, that's pretty much about where I hang. 45 degrees is getting a little bit hot, but 10 degrees is, in Fahrenheit, what is about 50 degrees or so. So, you might need a little jacket, a light sweater. These are a lot of bacteria that we think about. And then we have heat-tolerant bacteria that are either thermotolerant, they can handle warm heat, or they're heat-requiring. They're thermophiles. And we've got this term that we kind of talk about, hyperthermophiles, and these are the guys that grow in near boiling water. Now, what's really interesting is, if you think about temperature and the different macromolecules, temperature is going to have some pretty serious effects on macromolecules. Proteins and membranes, they begin to change their structure depending on the temperature. Let's just take proteins for example. Remember, they're being held together by the covalent bonds in their primary structure, which is not going to be bothered by really anything other than really oven level temperatures. But those weak ass secondary and tertiary bonds, those are the ones that begin to fall apart as the proteins denature, and those can happen. Those bonds don't hold together all that well, depending on the protein. Even like when you're running a fever, you begin to break them a little bit. So proteins begin to lose their structure at higher temperatures. Now, from another perspective, when things get cold, there are weak-ass bonds that typically wouldn't form at operating temperature, like myself, 37 degrees centigrade. But when you get them colder, they actually now can begin to form as the protein begins to kind of fold up and ball up a little bit tighter. And the protein may not work at lower temperatures. So another aspect of this that is a general truism is that as things get colder, there is just less free energy in the system. And remember, we talked a little bit about how enzymes work because they've got the energy of activation. They have to have a little bit of energy put into them in order to carry out their chemical reaction. And if things get too cold, that energy of activation just isn't sufficient. That's why putting things in the freezer is a good way of storing stuff. And so for our for humans, we are able to maintain our body temperature. And so as long as you're able to keep shivering, you have a jacket on, or you're able to keep sweating, you're maintaining your core temperature at around 37 degrees centigrade, or 98.6 or whatever. And that means that your proteins are maintaining their shapes. Now, organisms that don't have the ability to maintain their own body temperature, they are subject to the whims of the weather. So various kinds of what we think of as cold-blooded animals, animals that don't maintain their own internal body temperature, like snakes and various reptiles. They will exhibit basking behavior where they'll actually sun themselves, or if things start to get too hot, they'll go under a rock. So they are actually taking steps to maintain their body temperature. Now bacteria can't do that, so they don't really have the ability to move around quite like that, to hide under a rock. They are going to find themselves in conditions that they can grow in, or conditions that they can tolerate, and wait for things to get better. So another part of this is that the membranes, if they start to get too warm, they can get too liquidy, too fluidy, and they kind of just spread apart, as oils. If they get too cold, they get too rigid, and they can't fold and move around. And so the ability of a protein and membrane structures to maintain itself in these different temperatures is critical. Most organisms tend to be more or less set for a specific kind of conditions and can't handle a wide range of conditions. And most of those conditions tend to be sort of standard, temperatures you might find in the bulk of the environments on the planet. But there are bacteria everywhere you look on this planet. You're going to find bacteria. I mean, as we said, there's bacteria that are on the outside of the International Space Station. So bacteria can be awfully tough. And there are some bacteria that grow and thrive at very low temperatures, or even more bizarre, at really, really high temperatures. So that was just something to think about a bit. Now, if you recall, we mentioned the notion that you need to have a place to put the electrons when we're talking about the different aspects of nutrition. You need an electron source, but then you also need a place to put those electrons as you're carrying out the breakdown of things. So you need an electron source to build stuff, but you need a place to put electrons as you generate them when you're breaking things down. The standard kind of molecule that we use as our final dumping spot for electrons is oxygen. So you know this maybe from anatomy and physiology. You need to breathe, you need to keep breathing, and that's because oxygen is being used as a final electron acceptor. So we've got a variety of different bacteria that can actually maintain their living conditions in different conditions as well. So there are thinking about oxygen as a specific environmental condition. You wouldn't necessarily think about this, is that for you and I, oxygen ain't like an option. You got to have oxygen. Now, that's partly because we're multicellular complex organisms. We need a lot of energy. We dump a lot of electrons. We got to have oxygen all the time. These single cell guys, like yeast even, but certainly all bacteria, all prokaryotes, they don't need that much energy all the time. I mean, they can kind of just hang around. And so they actually are able to grow in a wide range of conditions. We've got oxygen as a number one condition to think about. First, there are organisms that are referred to as being obligate aerobes. And an obligate aerobe, it is obligated. It's got to have oxygen. So you have to have oxygen around in order to carry out cellular respiration and to generate ATP. There are bacteria that we think of as microaerophiles. They require oxygen, but in really very small amounts, much less than we do, and much less than an aerobic or an obligate aerobe would require. Then we've got organisms that can work without oxygen, and these are referred to as facultative anaerobes. And you can be a organism that typically uses oxygen, but you can live without it. And this is what results in anaerobic respiration, or also known as fermentation. So a lot of bacteria end up being facultative anaerobes, particularly bacteria on the cell surface or on your surface of your skin. And then we've got some bacteria like Clostridium botulinum, the bacterium that causes botulism. It is an obligate anaerobe. It requires the absence of oxygen in order to actually grow. Let's kind of think about that for a minute. They require a growth condition that has no oxygen around. They're killed by oxygen. They're making ATP either by photosynthesis or primarily by anaerobic respiration or fermentation. And this is actually a really interesting kind of phenomenon in that if you've got a puncture wound, that can be a pretty serious kind of wound compared to a scrape on the surface. The problem is that a puncture wound, you can end up with bacteria that were found in the soil and kind of just hanging out that are obligate anaerobes. They get in there. The other bacteria that came along with whatever it was that jabbed you, that nail that you stepped on, they began to consume the oxygen in that microenvironment of the tissue. The tissue begins to go anaerobic, and those facultative anaerobes or obligate anaerobes, they either die off or slow up. But now that the oxygen has been pulled out, we have the perfect conditions for some really nasty bacteria that are obligate anaerobes, and they're able to start growing. This happens in a variety of different tissues that don't get good oxygen supply, like your sinuses. If you get a really bad sinus infection, you can actually start to generate an area inside your sinuses that is anaerobic, that has got no oxygen. Puncture wounds can do this. If you think about this from a public health perspective, when you're canning foods, if you don't do like pumpkin, if you don't can it and heat it properly to kill off all the bacteria, you can have bacteria still in there. They begin to consume the oxygen. They probably wouldn't be particularly pathogenic bacteria, but once they consume the oxygen in that can, they now have established conditions that allow the really toxic bacteria that maybe only one or two were there to start growing. So that's some of the things that can happen. Now, the issue of this is there are some toxic byproducts of oxygen. And we don't really think about that so much because we use oxygen all the time. But all the time, our mitochondria are generating oxygen radicals, the O1. Oxygen is typically found as O2. And an O1, or 1/2 O2 as is often presented, is a really corrosive molecule. This is the active ingredient in hydrogen peroxide. And so you have molecules in this, in biochemical systems that are using oxygen that can generate peroxides and oxygen radicals. And these can be really damaging. Now, what we've got is a series of enzymes, bacteria and animals and eukaryotes, that can take the oxygen radical and begin doing things with it. Things like superoxide dismutase, catalases, and peroxidases. These are all generating different kinds of oxygen radicals and different sorts of highly reactive, highly oxidative molecules. And there are enzymes that can actually break these things apart and render them non-toxic. So superoxide dismutase is taking oxygen radicals and converting it to peroxide, hydrogen peroxide. Catalase can take hydrogen peroxide and break that down. And we can end up with different kinds of molecules. Glutathione peroxidase, just another term, don't need to memorize these, is also able to take peroxide, H2O2, and convert that into a non-toxic material, i.e. water. So, also when we're thinking about environmental conditions, we've got pH to concern ourselves with. So, we have, on our bodies, we've got a couple of different pH type environments. On your skin, you probably have a slightly lower pH. Saliva and tears probably have a pH of around 7 neutral. Inside your stomach, you've got extremely low pH. And then, of course, in the external environment, we've got areas of the environment that can be more or less neutral, but some areas actually can be extreme. And as we've been saying, you're going to find bacteria everywhere. That's one of the cool things to think about, is that any kind of phenomenally extreme environment you can think of, there are going to be bacteria there that are actually growing. So you go to the top of Mount Everest and it's 40 degrees below zero and phenomenally low atmospheric pressure. There are going to be bacteria up there that made it to the top, and they actually thrive up there. We go down to places that are carrying out mining that use, like ore mining, that use acids as a way of extracting the ore, and you end up with these large acid pools, and you'll find bacteria there. So we've got bacteria, most of them are working in the neutral range, around pH 6 to 8. These are referred to as neutrophiles. So if they get a pH above 8 or below 6, they start to have their proteins being denatured. Now, we've got some things that are in yogurt, like lactobacillus and Streptococcus thermophilus. Lactobacilli actually like moderately acidic environments. That's what they grow in. And they're not going to be growing in things that are extremely acidic, but they do work in low acid. So these are what we're referred to as acidophiles. They are loving acids in that they require a pH below 6. Generally, they don't go down too low. And this is this type of stuff that we find in things like sour cream, yogurt, sauerkraut, organisms that make vinegar, because that actually can be a bacterial fermentation process. Just one of the things to think about with this is that with yogurt, you have milk, and milk will go ahead and start to sour if you let it sit out too long. But if you inoculate it with different kinds of acidophilus, lactobacillus, different sorts of bacterias, you actually can have conditions in which you'll get bacteria, a lot of bacterial growth to make it, in fact, so thick that it's like yogurt. But it won't be super nasty like sour milk. And you'll actually begin generating a bacteria culture that actually starts to shift the pH down to allow for those bacteria to grow even better. With sauerkraut, you take cabbage and you add lots of salt, which inhibits a variety of different kinds of bacteria. The bacteria that are present, mainly things like Acetobacter and Lactobacillus, they start to convert the sugars in the cabbage into vinegars and lactic acids and different kinds of acids, and they begin to create an environment that is conducive to their growth, and it kind of blocks other things from growing. The same with fermented pickles and things like this. So there are some, though, that are extreme acidophiles, and they live below pH 4. Some are found at even pH 1, which is really acidic. I mean, that's like, very nasty stuff. And they're able to actually thrive there. We have gut bacteria that are operating in our stomachs that actually operate at very low pHs. As it turns out, there's not a lot of bacteria that tolerate a real high pH, but there are some, these basophiles. Now, a couple of last environmental conditions that we don't typically think about, and these are things like different levels of pressure and maybe even salt concentrations. So hydrostatic pressure is a condition. These are bacteria that actually can live under tremendous pressure very deep in the ocean. And so, there are submarines that are able to go phenomenally deep into the ocean, the Challenger being one of them. And they're able to get down very, very low to some of the deepest parts of the ocean, and they find bacteria there. As our slide says, we don't know why, and they're medically irrelevant. So let's move on. All right. Other conditions, though, that are important are things that we refer to as osmotic pressure. Now, and you probably have had this in, like A&P, I hope? I wonder. the idea of osmosis. Let's just kind of work through this for a moment. Osmosis is a phenomenon of having a plasma membrane, what we refer to as a semi-permeable membrane. It allows water to move through and non-polar molecules to move through, but charged molecules, ions like salts, can't move through. And so you end up with some interesting kind of phenomena going on. Mainly, water is going to move towards the area of higher salt, either inside the cell or outside the cell, depending on the conditions. This can generate osmotic pressure, either positive pressure, that is water moving into a cell begins to pressurize that cell, or a negative osmotic pressure, water moving out of the cell and starts to shrink the cell up. And we've got, of course, these terms. Isotonic conditions are salt concentrations in which the salt is equivalent on either side of a membrane, and water is flowing back and forth, diffusing, but it's not accumulating one side or the other. Now, the hypertonic, meaning tonic being salt, and hyper being high, hypo, like a hypodermic being low, hypertonic solutions have a higher salt concentration compared to their environment. And with higher salt concentrations, water always follows salt, so water starts moving into the hypertonic solution. And you're going to begin to accumulate water, so water starts moving in. In a hypotonic condition, we've got the concentration of salt is low compared to the external environment. Water follows salt. You start moving water out, you begin losing salt. So these kinds of conditions actually can influence bacterial growth. And there are some bacteria that prefer different kinds of salt concentrations. And so when we're growing bacteria, we typically grow them in a fairly salty beef broth. And it's actually saltier than I'd want to eat. But that is a condition that allows the bacteria to not lose water and not take up water. It is isotonic. If you take bacteria and put them into pure water, the bacteria are going to be hypertonic in relationship to the water. Water follows salt. Water begins moving into those cells. They'll pop after a while. They got a cell wall, so that'll help, but they can definitely start to swell up like a sick pup. The other side of this, if you put cells into a really high salt solution and they become hypotonic in relationship to their environment, they begin to lose water. They'll start to dehydrate, and this is what dehydration is doing. So there are conditions then. And there's some interesting things, though, with the bacteria. Because they do have their cell wall, they actually can manage a hypotonic environment in which they are themselves higher salt than the external environment. I guess, let me rephrase that. They can be hypotonic to their environment, in a hypertonic environment. So if we've got a hypotonic sort of system here, water is, all right, I'm screwing this up compared to the slide. We throw the cells into a hypotonic solution. That is a watery solution. Water starts moving into the cell, and like with red blood cells, because they don't have a cell wall, they begin to swell up, and they pop, right? That is cells in a hypotonic solution. Now, plant cells, bacteria cells, their cell wall, they begin to take up water, and they actually are going to pressurize. And so generally they require a slightly, like plants require a slightly hypotonic conditions. They like to be in conditions in which they've got a little bit more salt inside their cells than outside, and bacteria as well. And bacteria tend to live well in a slightly hypotonic environment, with the exception of bacteria that have been treated with antibiotics like penicillin, because that is going to break apart their cell wall or inhibit their cell wall formation, and now they're like a red blood cell. They pop as well. I just like making that popping sound. All right, an isotonic solution is really not too much of a problem. the cell wall isn't really needed to maintain cell structure. So a red blood cell is able to maintain its structure in an isotonic solution. That's why when you're giving somebody an IV, you're giving it to them in an isotonic solution. If you're giving somebody an IV that is pure water with whatever drug, you ought to check your labels or something. Don't do that. So you always give IVs in physiological saline. It's got physiological pH, and it's got about 0.9% salt. That is isotonic to our bodies. Now, what this means is the bacteria are probably going to be less turgid. They're not going to be pressing against the cell wall. They're going to be a little bit more flaccid, but they can survive. They're okay. That allows the animal cells to have some flexibility. Obviously, this is kind of the system that's involved in the kidney processing of urine. And so the kidneys actually are involved in regulating salt concentration. All right. Then we've got the other side of this coin, the third side, I don't know, middle, even one side, but regardless, the hypertonic solution, in which we got a really high salt concentration that you throw the cells into. You throw red blood cells into a hypertonic high salt solution, the water follows salt, water moves out, and that's going to start screwing up the cell concentrations. The cell will begin to shrink, not so good, makes your plants droop, they're sad, and the bacteria and fungi, they begin to shrivel up inside their cell wall. And what this means is they're going to die in a high salt solution, and they're going to end up actually not being able to survive very well. So sweat or sweaty, salty skin ends up being a hypertonic solution, and that's one of the reasons why bacteria are being knocked back from our bodies. Just one more protection system. So here's an interesting thing to think about in terms of you and I and bacteria, is that we present a fairly tough environment for bacteria to live on. Now, of course, they do. So our skin is relatively dry compared to like a frog or a fish. We don't have a slime layer on us. Our skin is fairly acidic. We've got fats and oils on our skin. That is going to be a protective mechanism to dissolve membranes and things. We have a fairly salty environment on your skin surface. Try licking yourself. Look if you're sweaty. That's going to be making a hypertonic environment. We've got then, of course, protective enzymes like lysozyme in tears that actually break apart cell wall. And one of the things when you're trying to pop cells, and as a molecular biologist working with bacteria to clone them, I routinely need to cause cells to break apart so I can extract the valuable proteins that they have produced. Lysozyme, typically from chicken egg white, is used to dissolve the cell membrane or the cell wall, and then they can pop very easily in a water solution, an aqueous, low salt solution. Otherwise, if you take bacteria and put them in salt, while they may not thrive very well, the cell wall won't pop, and you can't get at the proteins inside by just putting them in water. Even putting them in high salt doesn't really do it. You have to do something to degrade that cell wall. We're pretty adapted to prevent bacterial infection. It is amazing that bacteria can live on us at all, but they do. All right, we're going to stop here so we have a little bit of stuff to talk about on Friday, and this is just some real basic stuff of the kinds of things you need to grow basically the materials that we have in our bacteria media. And actually, this is sort of a nice little discussion there. We'll get to that a little bit on Friday. But that's what we're going to cover for our exam. And let's Zoom on Friday. As I said, we'll set up the exam for doing it next week. At this point, I'm going to set it up on Moodle where You'll have an hour to take the exam, but you can take it whenever you want. If anything, I'm flexible. I'd like to be able to throw my foot over my head, but I can't. Hey, you guys, as always, holler to me if you have questions or comments. I look forward to talking with you on Friday. And I'm Dr. G. I'm out. Peace.